Good evening, everyone. Welcome to BioTecNika's Vijeta Series. These series are specifically targeted to for CUET UG. CUET is Central University's entrance test, right? So BioTecNika is running these series every day from 28th June to 13th July. Time is 6 to 7 p.m. And I'm Dr. Nupur Nagvekar, and I'll be taking today's session for you, which is Unit 2. So, in Unit 2, the main heading is genetics and evolution, and within that, the topic that we are going to cover is Mendelian inheritance. Today, it's going to be Mendelian inheritance, and in the next session, we'll be talking about extensions of Mendel's principles. Right? So, are you all ready to start? Can we start the session now? If you're willing, let's directly get on to it. Yes. So, you see, principles of heredity were given by Gregor Mendel, right? That is why we know him as the father of modern genetics. Now, when he was working at the monastery, he conducted certain breeding experiments with pea plants. When he was conducting his experiments, that was around 1856 to 1863. That was the time period where he conducted his experiments with pea plants. He came up with his results, he came up with his conclusions, he made a paper on it, he even presented it somewhere around 1865. But unfortunately, at that time, the biological society, the group of people who saw his work, they could not understand the importance of the results, the conclusions that Mendel had given. At that time, nobody seemed to understand that what Mendel had concluded were actually the most basic principles of heredity and inheritance. So at that time, nobody understood the importance of Mendel's work. Later on, he stopped working on his research, he went into administrative work, and eventually he died in 1884. And even by that time, till that time, nobody had recognized Mendel for his contribution to genetics. Then around 15-20 years later, three different botanists independently started working on his experiments. They conducted similar experiments and they arrived at the same conclusions that Mendel gave, and that is when they understood the importance of his work. So they were the ones, Hugo de Vries, Eric von Schemark, and Karl Correns, they were the ones who brought the world's attention to his pioneering work. And today we know him as the father of modern genetics. So This topic basically tries to understand what work he did and what were his results and conclusions, and today what principles that we have based on his work, right? So Mendel's experiments, he chose the pea plant. Scientifically it's called Pisum sativum. So he chose the pea plant for his work, but that was not a random choice. He didn't just choose pea plant randomly. There were reasons for choosing this plant. Firstly, the plant is easy to cultivate, especially at that time where he was located. Pea plants were easy to cultivate. They are relatively rapid growing, okay, so not as fast as bacteria or Drosophila, but still compared to his the time where he was alive at that time, 1 year generation time was a good time. It was considered a relatively rapid growing organism. Also, one pea plant will produce many offsprings, right? So if there's one fertilization event, it will give rise to pea pods, and pea pods will have pea seeds. So you will get many pea pods and pea seeds from one pea plant, so it will produce many offsprings. And that is why you will understand that Mendel, when he gave the results, you can find that his ratios were so sturdy, as in the numbers that he got from his experiments were in large quantities, and that is why he could give us those ratios of 3 is to 1 and 9 is to 3 is to 3 is to 1. So this helped Mendel to study the ratios of traits in progeny. Also, the monastery where he was working, there were farmlands nearby, and he had access to a large number of varieties of peas. They were readily available to him. Now, what Mendel did was he chose 7 characteristics for his study. Again, the 7 characteristics were not randomly chosen, okay? He chose specifically those characteristics that did not show a range of variation. Now, what do you mean by this? For example, the color of our skin, right? Color of human skin. It shows a range of variation. You can't say it's just white or brown. No, you can't say that. Our skin shows a range of variation. So Mendel avoided such characteristics. He chose 7 characteristics that were in two easily differentiated forms, right? For example, the characteristic is the shape of the seed, right? We all have eaten pea seeds, right? So we know this. So seed shape is a characteristic. Mendel chose two variants in this particular characteristic. So what were the variants that he chose? He chose pea plants that were producing round seeds, and he chose pea plants that were producing wrinkled seeds. So this is a characteristic, and these are the variants for that characteristic. Then the second characteristic was seed color. In that, the two variants that he chose for his experiments, that were yellow color and green color. The third trait was the third characteristic was pod shape. Here you can see this is an inflated pea pod and this is a constricted pea pod. Then the fourth characteristic was the color of the pod. The variants were green and yellow. The fifth one was the color of the flower. Here, the two variants were purple flowers and white flowers. The sixth one was the location of the flower. So, where is the flower located on the pea plant? It could be here, axial, towards the axis, basically throughout the axis of the plant, and terminal, that means towards the top or towards the tip, right? So, axial or terminal. And the size of the plant, that was the last characteristic or the seventh characteristic. That means a tall pea plant or a short or a dwarf pea plant. So like that, Mendel chose 7 characteristics and he wisely chose them. He chose characteristics that did not display a range of variation, but instead he chose those characteristics which show two easily differentiated forms, like round versus wrinkled, or inflated versus constricted, or yellow versus green. On top of that, he also adopted an experimental approach and he interpreted his results using mathematics. So he used mathematics, he used statistics to basically draw his conclusions and interpret his results. So that is what gave him an edge over others. Also, luck too slightly played a role in his success for his experiments, because luckily the characteristics that he chose, they were not linked. Okay, they were on different linkage groups, so that is why they did not interfere with his very nice ratios of 3:1 and 9:3:3:1. Now, before we go ahead, let us look at some important terms that are very commonly used in genetics, right? So, we just now spoke about characteristic. What's a character or characteristic? It is an attribute or a feature. Like, for example, the seed, the shape of the seed or the color of the seed, it's a character for that organism. It's a characteristic for that organism. So, it's an attribute or a feature. What is a gene? A gene is a genetic factor. It's a region of DNA that helps determine the characteristic. So if the characteristic is the shape of the seed, then there would be corresponding gene or genes which are helping determine that characteristic, which are helping in expression of that characteristic. So gene is a genetic factor. It's a region of DNA that determines the characteristic. What are alleles? Alleles are alternate forms of a gene, right? For example, here, these two are homologous chromosomes. Why do we call them homologous chromosomes? Because these two chromosomes are similar, and one chromosome has come from the maternal side and the other chromosome has come from the paternal side, right? So here at this locus, there are two alleles. Now, these two alleles could be the same or these two alleles could be different. If the alleles are same, you call them homozygous. Now, can you see here? This is allele capital R and this is also allele capital R. So, the alleles are the same. So, it is called homozygous. And on the other hand, it could be the recessive allele also. So, here allele is small r, here allele is small r. So, still it is called as homozygous. This is called as homozygous dominant. This is called as homozygous recessive. Now, what if the two alleles are different? What if the two alleles on the homologous chromosomes are different? If they are different, then you call it heterozygous. Right? If the two alleles are different, you call it heterozygous. For example, here, this is allele capital R, this is allele small r. The two alleles are different. That is what makes it heterozygous. So, what is an allele? Allele is alternate forms of the gene. What is locus? Locus is that specific location or that specific place on the chromosome where the allele is present. So, here, this is the specific location or locus where the allele capital R is present. This is the specific locus where the allele small r is present. Here, the allele capital R and small r are present. What is genotype? Genotype is the set of alleles that an individual organism has. For example, this organism has these homologous chromosomes. So what can we say about the genotype of this individual? This individual has the genotype capital R, small r, right? That is how we write the genotype. Here, for this organism, the genotype is capital R, capital R. For this one, it is small r, small r. So, what is genotype? When you list out the set of alleles, when you write the set of alleles that the individual organism has, that is genotype. So, this is genotype, this is genotype, this is genotype. What is phenotype or trait? Phenotype is basically the appearance of the character. This is genotype. But how the genotype manifests itself, how it shows in that organism, right? How it comes out in that organism, that is phenotype. For example, this particular pea plant will produce round seeds. Because the genotype is capital R, small r, the phenotype is round seeds. This one is homozygous dominant. The genotype is capital R, capital R. But what will be the phenotype? The phenotype also will be round seeds. And here the genotype is small r, but the phenotypically, how will be the expression be? It will be wrinkled seeds, right? So whatever be the genotype, how it manifests itself in the form of the characteristic, that is called as the phenotype, the appearance or manifestation of the character. Okay. And homozygous and heterozygous, we already discussed. What is homozygous? An individual organism possessing two of the same alleles at the locus. So if both the alleles are the same, then you call it homozygous. If both the alleles are different, then you call it heterozygous. So these are some important genetic terms that you know before we go on to the actual topic. Now, let's look at the monohybrid crosses. So, Mendel did monohybrid and dihybrid crosses. So, Mendel started with about 34 varieties of pea plants, as in he started with a lot many pea plants, but he spent about 2 years selecting the varieties that he would use in his experiments. He made sure that he is actually selecting homozygous plants, or plants with the homozygous genotype, for his crosses, for his breeding experiments. How did he do that? By growing the plants for about 2 generations. If you grow them for about 2 generations and there is no variation in the progeny, you are confirming that the offspring is same as the parent. That means you are confirming that it is a pure breeding variety. In other words, you are confirming that the plants are homozygous for that particular trait. So he carried out different crosses between different varieties. Now, you know that pea plants, pea flowers are bisexual. That means they have both the male part as well as the female part. You can see in the diagram over here. These are the male parts, the anther, the filament, and here is the stigma, style, and this is the ovary, and inside you can see the ovules, right? So both male and the female reproductive organs are present on the same flower. So pea flowers are bisexual. Now, normally peas are self-fertilizing. Self-fertilizing means in the same flower, the pollen released by this anther will land on the stigma and it will bring about fertilization in the same flower. But Mendel did not want that. Okay, Mendel wanted to control the fertilization. So what he did was, Mendel basically removed the anthers. If you remove the anthers, that means you are removing the male part of the plant. So he deliberately made the flower into a female flower. How did he convert a pea flower into a female flower? By removing the anthers, by cutting off the male parts. And then also what he had to do was, he had to bag the stigma. He had to put a sort of a bag on the top of the stigma, so as to make sure that random pollination does not occur, and to be sure that the cross is from his selected plants, the cross is from his selected flowers. He used a brush like structure to just tap on the male flower anthers and tap it onto the stigma, though, so that it is a very specific cross, so that specifically the pollen of his choice lands deliberately on the stigma of his choice, and that is how he brought about his crosses, right? So when he did the cross, the what is the products of pollination and fertilization in pea plants? That is the pea pod and the pea seeds, right? The ovule will develop into the pea seeds and the ovary will develop into the pea pods. So when you plant these seeds, each seed will develop into a new plant. So that is how he got the progeny from his crosses. So let's look at the crosses that he carried out. The first one being the monohybrid crosses. What are monohybrid crosses? Mono indicating one. So if you choose organisms, male and female organisms, that differ with regards to one single contrasting trait, that means you are studying one single contrasting trait in that organism. So one trait which is varying between the male parent and the female parent, that is when you call it a monohybrid cross. For example, Mendel crossed a pure breeding homozygous pea plant for round seeds with one that was homozygous for wrinkled seeds, right? So he crossed plant which was producing round seeds with plant that was producing wrinkled seeds. So what is P generation? P generation is the parental generation. It is that generation which is undergoing the cross, which is being crossed. P generation, after undergoing the pollination, the fertilization process, the next generation that is formed, that is the offspring of the P generation, is called as the F1 generation. F1 standing for filial one. Filial one generation is nothing but the offspring of the P generation. So like that, if the F1 generation is allowed to be crossed, if fertilization is allowed in the F1 generation, then the offspring of the F1 generation will be called as the F2 generation, indicating it is filial 2, right? It's the offspring of the F1 generation. So like that, you have the nomenclature for the following generations. So after crossing the two varieties in the parental generation, Mendel observed the offsprings that resulted from the cross. Okay, let's look at his cross. In his monohybrid cross, what is the parental generation? He crossed pea plants which are producing round seeds with the plants which are producing wrinkled seeds, right? So when he did this cross, he observed the pea plants of the F1 generation. He found that all the progeny in the F1 generation are all producing round seeds, right? This was his major basically identification, that all the seeds that are being produced in the F1 generation are round seeds. Then what he did was he allowed the F1 generation to self-fertilize. That means from the same flower, the same anther will be used to pollinate and fertilize the female parts of the same flower. That is self-fertilization. So he allowed self-fertilization of the F1 generation. And what did he find? In the F2 generation, so this is the next generation now, the progeny of the F1 generation is called as the F2 generation. In the F2 generation, he found that both round and wrinkled seeds were produced. That means there were plants which are producing round as well as wrinkled seeds. So what basically can we conclude here? Because in the F1 generation, the round seeds were produced, and in the F2 generation, both the characteristics started showing itself. Both the traits started showing itself, round and wrinkled, but specifically in a ratio of 3:1. Okay, he found that in the F2 generation, the round seeds were about 3 parts and wrinkled seeds were about 1 part. So these were the actual numbers found out by him: 5474 round seeds and 1850 wrinkled seeds. So Mendel found that the F1 generation expressed only one of the phenotypes, that is the round phenotype of the parental generation. All the F1 seeds were round. He did multiple such crosses, so he did not stop at one cross, and he did not give his results after just performing one cross. He tried multiple such crosses for this trait and for the other characteristics also, for the height, for the color of the flower, all the 7 characteristics. He did the crosses multiple times, and all the time he got the same result. What was the result? That in the F1 generation, one of the two variants is expressed. Okay, so if your variants are round and wrinkled, only one of them would be expressed in the F1 generation. So from these F1 seeds, he planted the F1 seeds. So now a new plant arises, the seeds will germinate, seedling will form, and the seed will produce a new plant, right? So he planted those seeds, cultivated those seeds, and he allowed these F1 generation plants to self-fertilize. That gave rise to the next generation, that is the F2 generation. Here he found that both the traits emerged, emergence of round as well as emergence of wrinkled. He counted 5474 round seeds and 1850 wrinkled seeds in the F2 generation, and that is how he came to the ratio of 3:1. So, the presence of both round and wrinkled seeds in the F2 generation can only be explained if the F1 plant has both the genetic factors. If the F1 plants, these plants, okay, if the F1 plants have the genetic factors for round as well as wrinkled, only that explains the appearance of wrinkled seeds in the F2 generation. Otherwise, how wrinkled seeds will show themselves in the F2 generation? That means they are there here. The genetic factors for wrinkled seeds are there in the F1 generation, but they are hidden. They are hidden, they are not expressing themselves, but in the F2 generation you can see them, they appear, right? So Mendel concluded that each plant must therefore possess two genetic factors which encode a character, and today we call these genetic factors as alleles. So that means let's see how we can write this same cross but with the genotype. Now let's understand how the genotype is playing a role over here, right? So this is the parental generation, and it's the same cross that we saw earlier. This is homozygous round seeds, capital R, capital R. This is the plant producing homozygous wrinkled seeds. The genotype is small r, small r. Now, what happens during seed formation? That means the gamete formation process. In the gamete formation process, all the gametes produced by this homozygous plant contains the capital R allele. So 100% of gametes produced by this plant is having the capital R allele. And here 100% of the gametes produced by this plant have the small r allele. So, in the process of fertilization, what happens? In the process of fertilization, the male gamete fuses with the female gamete, right? That is how the zygote is produced. So, in the process of fertilization, the gamete with capital R fuses with the gamete with small r, and that is how you get the zygote producing the next generation, that is the F1 generation. So this generation, it manifests itself, the phenotype that it manifests is round seeds. But genotypically, how is it? What is the genotype of the F1 generation? The genotype of the F1 generation is capital R, small r. So this one and this one coming together, this is heterozygous. The F1 generation is heterozygous, right? So why is it not showing itself as wrinkled? Why is phenotypically it is round? Because though it is heterozygous, it has both the alleles. If it is heterozygous, that means it has both the alleles, capital R as well as small r. But why is it not expressing as wrinkled? Why is it expressing as round? That's because the dominant allele is the allele for roundness, and the recessive allele is the allele for wrinkled. So capital R is dominant over small r. And when both these alleles are present together, the allele for wrinkled does not express itself. And that is why in the phenotype, you only see the round expression. Only round is expressed, right? So when two gametes, one from each parent, fuse to form a zygote, the allele from the male parent unites with the allele from the female parent to produce the genotype. Those traits that appear unchanged in the F1 heterozygous offspring are dominant because round is expressing itself. So round is dominant over wrinkled. Now, what about the F2 generation? When F1 generation forms gametes, now what is going to happen? In the gamete formation process, the two alleles are going to separate, right? So the male parent produces two types of gametes. 50% of the gametes will have capital R, 50% of the gametes will have small r. Similarly, the female parent also produces 50% capital R and 50% small r. So now, since the F1 is allowed to self-fertilize, let's look at the progeny of the F1 generation, which is called as the F2 generation, filial 2 generation. That's the offspring of F1. Now, what combinations do you have? All the combinations that are possible. Capital R joining with capital R. Okay? Capital R joining with small r joining with capital R, and small r joining with small r. These are the four combinations that are possible. Okay? Capital R, capital R, capital R, small r, small r, capital R, small r, small r. But since these two are the same, let's club them together. Okay? These two are the same, right? Both of them are heterozygous. You've just changed the order, but they are the same. So, in the F2 generation, what is found phenotypically? Phenotypically, how many round and how many wrinkled do you get? You get 3 round. Okay, 1, 2 and 3. That means this is capital R, then this is capital R, this is small r, capital R. 3 parts, 3 out of 4 are round and 1 out of 4 is wrinkled. But what about genotypically? Genotypically, how do you write this? Genotypically, if the dominant alleles come together, then this parent, this particular plant is homozygous dominant. These two are heterozygous, and this one is homozygous recessive. So when dominant and recessive alleles are present together, the dominant allele shows itself phenotypically and the recessive allele is masked or it is suppressed. Mendel also allowed his F2 generation to self-fertilize. That means he also allowed F2 generation to produce F3. And what he found was, if this is allowed to self-fertilize, if homozygous dominant round is allowed to self-fertilize, you will only get round in the progeny. Okay, only round seeds are produced because that is homozygous dominant. Similarly, if wrinkled is allowed to self-fertilize, it will only produce wrinkled seeds in the F3 generation. But if heterozygous seeds, if these heterozygous plants are allowed to self-fertilize, again in this you will find round to wrinkled in the genotypic ratio, of 1:2:1 or phenotypic ratio of 3:1. 3 parts of round and 1 part of wrinkled. But if you look at the genotype, why is the genotype 1:2:1? It's because one part is capital R, okay? Then 2 parts are capital R, small r, heterozygous. And again, there is one part which is small r. So that is why the genotypic ratio is 1:2:1 and the phenotypic ratio is 3:1. So what were the important conclusions by Mendel? Each plant must possess 2 genetic factors to encode a character. The 2 genetic factors separate when the gametes are formed. One genetic factor goes to each gamete. When dominant and recessive traits are present together, the dominant trait expresses itself and the recessive trait is masked. Separation of the genetic factors into gametes occurs with equal probability, and pairing of the gametes is also random. Now, what do you mean by this? Both these parents are heterozygous, right? This is capital R, small r, this is capital R, small r. They are heterozygous. Each one will produce half of the gametes with capital R allele and half of the gametes with small r allele. Both the parents will do that. Half of the gametes with capital R allele, half of the gametes with small r allele. Now, how do the gametes pair with each other? It's a random process. So, the separation of genetic factors into gametes, it occurs with equal probability. But during the fertilization process, the pairing is also random. Okay, so capital R, small r, capital R, small r. These are the four gametes. This is from the male parent, this is from the female parent. So while pairing, the pairing can be random and it can be with equal probability. So capital R can fuse with capital R, right? But with the equal probability, capital R can also fuse with small r, like this. So this small can go with this capital, right? And this small can go with this small r, small r. So that is what it is. So pairing of gametes is also random. So the following genotypes are produced in equal proportions among the F2. Capital R, capital R, capital R, small r, small r, capital R, small r, small r. But since these two are the same, you club them together. So this is one, this is one, this is one, and this is one. But you club these two together, and that is why you get the ratio of 1 is to 2 is to 1. So this particular conclusion has been now formalized into the principle of segregation and the concept of dominance. What is the principle of segregation? It states that each individual diploid organism possesses two alleles for any particular characteristic. So we are all diploid organisms. Diploid organisms will have two alleles for any particular characteristic. These two alleles will segregate or separate during the gamete formation process, right? If it is heterozygous, then capital R and small r will separate into different gametes because these two chromosomes are going to separate. This chromosome will go to one pole, this chromosome will go to the other pole. So there is separation of alleles in the gamete formation process. Furthermore, the two alleles segregate into gametes in equal proportion. So that is why we have 50% capital R gametes and 50% small r gametes. The division is in equal proportions. What is the concept of dominance? The concept of dominance states that when two different alleles are present in a genotype, like for example this, the genotype is capital R, small r. So the two alleles are different. In that case, only the trait encoded by one of them, that is the dominant allele, is observed in the phenotype. So that is the reason why if this is the genotype, you see the phenotype as round. Why do you see the phenotype as round? Because the dominant allele is expressing, it is the one which is dictating the phenotype, and the recessive allele, small r, is hidden. It is suppressed. It is not expressing itself. All right. Now, before we go to dihybrid, let's quickly look at what is test cross and back cross. In test cross, you're testing something. The name test cross is because you're checking for something, you're testing something. And what is that? What is it that you're checking? You're trying to determine the zygosity of that individual. What is zygosity? Now, let me explain. This is a round seed, right? This is a round seed, and you can clearly see that this is a wrinkled seed. Now, if it is a wrinkled seed, you can say with surety that the genotype is small r, small r. But if there is a round seed in front of you, can you say with surety what is the genotype of that round seed? Can you be very sure just by looking at the round seed? Can you say with surety what is the genotype? No, right? Because this particular round seed, it could be homozygous dominant or it could be heterozygous. That means the genotype of this seed could be capital R, or it could be capital R, small r. You can't say that just by looking at that seed. So you don't know what is the zygosity. Is it homozygous dominant capital R, or is it heterozygous capital R, small r? You can't make that out. So that is what you're testing. You're trying to determine the zygosity. So in this test, what do? You cross that individual whose zygosity you're trying to determine, you cross it with a phenotypically recessive individual. Right? So who is a phenotypically recessive individual in this case? It is this one. Phenotypically recessive. That means it's a wrinkled seed, a plant which is producing wrinkled seeds. So when you do this cross, when you do this cross, you look at the progeny. You look at the F1 progeny. Based on what is the outcome of the F1 progeny, you can say with surety whether it is homozygous dominant, whether this plant is homozygous dominant, or whether it is heterozygous. Okay, how can we do that? Let's look at the example. So to determine if the phenotype round seeds is homozygous or heterozygous, you have to cross it with a plant producing wrinkled seeds. If the genotype of this parent is homozygous dominant, this one is obviously recessive, small r. What do you get in the F1 progeny? The F1 progeny seeds will be all round, 100% of them. All round. There is no chance of wrinkled at all. 100% all of them will be round because the genotype will be heterozygous. Okay, so in the F1 progeny, if you are getting all round seeds, with surety you can say that this particular parent is homozygous dominant. And what will happen if this is heterozygous? If this parent is heterozygous, what is the F1 progeny that you will get? You will get round and wrinkled in 1:1 ratio. In the F1 progeny, you will get round seeds and wrinkled seeds in 1:1 ratio. Okay, so that is how you carry out the test cross. What is back cross? Back cross is carried out in basically horticulture or in animal breeding. Back cross is different. The purpose of carrying out back cross is different. In back cross, the purpose is to achieve an offspring with a genetic identity which is closer to that of the parent. Okay, for example, this is a bull of breed A. Let's say you've brought this somewhere from abroad. Okay, it's some other breed, you've brought it from outside. This is like a desi breed cow of breed B. So when you cross these two, you get the F1 female progeny who is having the genome content of both the bull of breed A and the cow of breed B. But in her genetic content, you want more percentage of the bull of breed A. That means you want more genetic content of breed A. So what you do is you cross this female progeny with one of its parents, that is the bull of breed A, or another organism who is genetically similar to the bull of breed A. Right? So when you do this cross, the progeny that you get has a higher percentage of breed A genetic content in their genome. Yes, now it's 75% A and smaller percentage of B, right? So this actually gives you some desirable characteristics, maybe more milk production or more meat or better fat content or whatever, but it gives some advantage to doing these crosses. So these are back crosses. Back crosses are different from your test crosses. Now, Punnett square is a way by which you can determine the outcome of genetic crosses. To make a Punnett square, there are two steps. The first step is to identify what gametes are produced by each parent. Let's look at the example. You have a heterozygous parent being mated with a homozygous parent. A heterozygous tall plant is being mated with a homozygous dwarf plant, right? So with this information, first you identify the gametes. If it is heterozygous tall, it will produce two types of gametes, capital T and small t, in 50-50% proportion. And if it is homozygous small, that means if it is homozygous dwarf, all 100% of the gametes produced will have the dwarf allele. Once you have identified the gametes, you just place those gametes on the top and the side of a grid. So you have to make a grid like this. Okay, you have to make a grid like this and you place the gametes on the top and the side. So these gametes are from one parent and these gametes are from the other parent. So for example, see, this is the tall plant, produces 50% capital T, 50% small t. This is the dwarf plant, all 100% are small t. So the dwarf alleles you have written over here and the heterozygous gametes you have written over here. Now just write the combinations in each of the square. So this is capital T small t, capital T small t, small t small t, and small t small t. That means you are getting tall and short progeny in 1:1 ratio, and the genotypic ratio is also 1:1, because 50% of them are heterozygous, capital T small t, and 50% are showing homozygous recessive, small t. So this is a Punnett square. The last part is dihybrid crosses. In monohybrid crosses, you consider one trait which is different in the parents, right? In dihybrid cross, you consider two traits which are different in the parents. For example, you consider seed color and seed shape, right? These are two different characteristics altogether. Now, seed color has two variants, yellow and green, and seed shape has two variants, round and wrinkled, right? So now what you do is, what Mendel did was, he took homozygous P with seeds that were round and yellow. Okay, so he took one parent who was producing round and yellow seeds. Genotypically, we write it as capital R, capital R, capital Y, because it's both the characteristics are dominant. Round and yellow both are dominant. Whereas the other parent that he considered had wrinkled and green seeds. So wrinkled is recessive to round and green is recessive to yellow. So genotypically you write it as, I mean the genotype of this parent you write as small r, small r, small y, small y. So now we carry out the cross. First, identify the gametes produced by both the parents. This parent is producing only one type of gamete. All of them have capital R, capital Y alleles. This parent is producing one type of gamete. All 100% of the gametes have small r, small y. And when they are allowed to fertilize, and when you allow the fertilization to take place, these two gametes will fuse together and you will get capital R, small r, capital Y, small y. So what is this? This is heterozygous. Okay, F1 generation is heterozygous. How does the F1 generation now produce gametes? The gametes produced by the F1 generation are capital R, capital Y. You know, that is one combination. Then capital R, small y, that is another combination. Small r, capital Y, that is another combination. And small r, small y, that is the last combination. So there are four types of gametes that are produced by the F1 generation. Now F1 generation is allowed to undergo self-fertilization. That means this heterozygous male is also mated with heterozygous female. So now all the gametes, now you have to make a Punnett square. Write all the gametes on the top and on the left of the grid. Once you've written the gametes on the top and the left, just fuse them. You know, each square represents the fusion of the gamete written on the top and on the left. So like that, now you have 16 such squares, right? 16 such squares represent all the possible combinations. You will find that in this in these F2 generation plants that are produced, about nine of them are round and yellow. Okay, these ones are round and yellow. Then there is one which is wrinkled and green. This one, there is only one which is wrinkled and green. Then there are three of them which are round and green. Which are those? These ones. See, round and green. And then there are three which are wrinkled and yellow. Right? So what is the phenotypic ratio for the dihybrid cross? 9 of them are round and wrinkled. 3 of them are round and green. 3 of them are wrinkled and yellow, and 1 of them is wrinkled and green. So the ratio that you get is 9:3:3:1. The ratio which was 3:1 in the monohybrid cross, for dihybrid cross it is 9:3:3:1. Right? So from the dihybrid cross, we deduce the principle of independent assortment. What is independent assortment? When F1 plants are allowed to self-fertilize, the alleles separate and one allele goes to each gamete. Yes? There's going to be separation of alleles. But when the alleles separate, they can separate in two different possibilities. This is possibility number 1 and this is possibility number 2. Okay, so you see here, this is one homologous pair, this is another homologous pair. This is one homologous pair, this is another homologous pair. The first possibility is that during meiosis 1, when the homologous chromosomes separate, the first possibility is that small r and small y go to one pole and capital R and capital Y chromosomes go to the opposite pole. Okay, this is one possibility. So small r, small y, this is one type of gamete that is produced, and capital R, capital Y, this is another type of gamete that is produced. But the other possibility is that during meiosis 1, when the homologous chromosomes separate, the other possibility is that small r can go with capital Y. You know, small r and capital Y chromosomes travel to the same pole, and the gametes have the combination of small r, capital Y. And on the other side, capital R and small y travel to the opposite poles. So the gamete that is produced is capital R, small y. So there are two equally probable arrangements of chromosomes in meiosis 1, right, during metaphase 1 of meiosis 1. And that is why you have these four types of gametes that are produced: capital R, capital Y, small r, small y, small r, capital Y, and capital R, small y. So capital R separates with capital Y and small r separates with capital Y. That is one possibility. Capital R separates with small y, small r separates with capital Y. That is another possibility. So this gives you the principle of independent assortment. This tells us that the alleles at each locus, they separate independently. The separation of the alleles at the R locus has nothing to do with the separation of the alleles at the Y locus. Okay, so that is why it is called as independent assortment. Both kinds of separation occur equally, and that is why all the four types of gametes are produced in equal proportions. These four gametes are produced in equal proportions, 1:1:1:1 ratio. This is principle of independent assortment deduced from dihybrid crosses. Okay, so let us quickly look at 3 questions and then we will stop for the day. Okay, so if you performed a monohybrid cross to investigate seed shape and obtained 650 round seeds in the F2 generation, approximately how many wrinkled seeds would you expect to have? Now the question says you have performed a monohybrid cross. You're checking for the shape of the seed. That is the characteristic that you're looking out for. And in the F2 generation, you got about 650 round seeds. Yes. Now in this, if 650 round seeds are there, then in the F2 generation, how many wrinkled seeds would you expect to have? Well, this is a monohybrid cross. In a monohybrid cross in the F2 generation, the ratio of round to wrinkled is 3:1. Isn't it? So if three parts are corresponding to 650, then one part corresponds to how much? The answer you should get is 217. Okay, the answer you should get is 217 because 3 parts correspond to 650. So one part will correspond to how much? Your answer should be 217. Next question. Imagine you're performing a cross involving seed color in garden pea plants. What F1 offspring would you expect if your homozygous parent producing green seeds with homozygous parent producing yellow seeds? So you're performing a cross in which you're testing for seed color. You have crossed homozygous parent producing green seeds with homozygous parent producing yellow seeds. So this is the genotype of the dominant parent. This is the genotype of the recessive parent. So yellow color seed is dominant over green. So what progeny would you expect in the F1? In the F1, see, this produces gametes with small y allele. This produces gametes with capital Y allele. These two will fuse together and you will get capital Y, small y. So 100% of them, all 100% of the seeds produced will be yellow. So the answer here is option B. The answer is option B, 100% yellow seeds. And the last question for the day. In order to find out if the tall pea plant is homozygous or heterozygous, it is crossed with a dwarf pea plant with the genotype TT. So such a cross is what? Here you are testing something. You're testing, you're trying to determine the zygosity of that plant, right? Whether it is homozygous or heterozygous. That is why you want to cross it with the recessive parent, the plant which is showing the recessive trait, the dwarf pea plant. So such a cross is called as a test cross, where you're testing the zygosity. All right. So guys, with this we've come to the end of Mendelian inheritance. In case you have any doubts, you can email it at this uh, uh, this thing ID. You can contact BioTecNika's toll-free numbers. You can post your testimonials here, and you can also download the BioTecNika app to get the latest information. All right. Thank you so much. In the next CUET UG series, we will also look at extensions of Mendel's principles. Thank you so much. Bye-bye. Have a great day.